Greetings, Pencrest High School students. It's Mr. Del Prado here. You don't see my face because I'm going to try to turn into a math teacher a little bit today. The beginning of our genetics unit has lots of conceptual ideas in it when we talk about alleles, dominant alleles, and recessive alleles. Um, but lots of times students get caught up in those details and then just want to have the basic idea of how to do a Punnett square. So I was going to show you where we get the letters from in a Punnett square, how to set up a Punnett square, and then how to interpret a Punnett square. So we're going to go all the way back to Mendel's cross with his pea plant. So if you remember, for the colors of the pea plants, there were two different colors. There was a purple color and there was a white color. Okay, so remember that those would be alleles on a gene. And remember that each individual gets two alleles, one from mom and one from dad. So if you remember Mendel's original plants, then he had a pure purple plant which had two dominant genes. He got that by self-fertilizing the plant over and over and over again. Okay? And then he crossed it, and that little dot there is like a multiplication symbol, meaning like he crossed that plant, right, with a white plant which had two of the recessive alleles. Again, if you want to know where we got the letters from, Mendel self-fertilized purple plants over and over and over again until he was reasonably sure that it had two of the same dominant factors on it. So there's one individual there. Okay? And he did the same thing with a white plant where he self-fertilized that white plant over and over and over again. And you can see the two genes there because that plant, again, has two recessive genes. Now, what you could do when you cross these plants together is you could look at all of the possible ways in which these letters turn into essentially gametes. So watch. If I make this individual the male and this individual the female, watch what we can do. And I'm going to draw little sperm cells here to make you feel uncomfortable, right? Okay, but there's one dominant gene. There's another one. Remember that these genes separate during meiosis. So your sex cells only get one copy of each gene. So there's our individual. There's the sex cells, okay? This big P goes here. This big P goes here. We'll make this the female. Here's the egg cells, okay? And again, through meiosis, we get a little P here and a little P here, all right? So now, look, here's what we could do. You can look at all the possible combinations of sperm cells and egg cells for offspring. If this sperm cell fertilized this egg cell, you would end up getting a baby or an offspring that has one dominant allele and one recessive allele. Remember, we say that that's heterozygous. This would be a purple pea plant. Maybe this sperm cell fertilizes this egg cell, right? And if that happens, you would get one dominant allele, one recessive allele. Again, that would be a purple pea plant. Okay? Well, maybe this sperm cell doesn't make it at all. Maybe it's this one that makes it. Okay? If this sperm cell fertilizes this egg cell, look what we get. One dominant, one recessive allele. All right? Again, this would be a purple plant. Or, here's our final combination. This sperm cell fertilizes that egg cell. Okay? And then we end up getting, again, another heterozygote. Every single one of these offspring would be purple plants. And that's exactly what Mendel discovered when he made it the true breeding purple plant with the true breeding white plant. He got all purple offspring. Now, there is an easier way to do this instead of drawing all of these arrows, and that's what I was going to show you. Okay? So we're going to still keep our parents up there. One of them is pure purple, one of them is pure white. But instead of drawing the egg cells and sperm cells, scientists have created a little tool to help us better see what the results of a cross would be in terms of the offspring, in terms of the babies. And that's the Punnett square. I always think it's a square, and I make four little windows. All right? I'm going to take this individual here, and I'm going to separate out the alleles and put them at the top of the Punnett square. Okay? I'm going to take this big P, I'm going to put it right here. I'm going to take this big P, I'm going to put it right here. And I'm going to do the same thing with the second individual, except I'm going to put it on the side. It does not matter if this one goes on the side or if this goes on the top, as long as you separate out the alleles. This is a little P, I'll do that. This is another little P, I'm going to do that. And all I do with the Punnett square is I essentially multiply the genes together. All right? Always put the capital letter first. You'll see why that's important when we get into some more complicated problems in genetics. I see a dominant allele and a recessive. Look what I got. Big P, little p. That's a heterozygote. Dominant and recessive, another heterozygote. Dominant, recessive, another heterozygote. Dominant, recessive, another heterozygote. All I did was essentially just multiply the genes together. You multiply the top genes by the genes on the side. Okay? Big P, little p, I get that there. Big P, little p, I get that there. Big P, little p, and big P, little p, and I get that there. Look what those are the genotypes. Each one of those individuals is heterozygous. What is the phenotype? What do those flowers look like? 100% of the offspring would be purple. Okay? And that's how you set up a Punnett square. But wait, kids, there's more. What Mendel did after that is he then crossed the purple offspring together. So he took one of these offspring and he mated it with one of these offspring, right? And this gets weird and a little bit creepy, but we're talking about plants here. So now, instead of me drawing the sperm cells and egg cells, I'm going to use these two individuals and I'm going to cross them together. So watch what happens. Here's my one individual here. It's a heterozygous, it's a purple flower. And I'm going to cross it with this one. It doesn't matter which ones I would have taken. They all have the same genotypes. And I get this. Now watch. Instead of drawing all those sperm and egg cells, I'm simply going to set up a Punnett square so that I can see what the offspring would look like. Ready? Here's my square. Let's make sure you can see that. Yep, it looks good. I'm going to take this dominant gene. I'm going to put it right there. I'm going to take this recessive gene. I'm going to put it right there. That's showing me the sex cells of parent number 1, which is right here. Now I'm going to do the same thing over here for parent number 2. I'm going to take this dominant gene. I'm going to put it right here. I'll take this recessive gene. I'll put it right here. That's parent number 2. I know these P's look similar to each other, so I'll try to make this one a little bit bigger. Okay. Now I do the same thing. Oh, I'm sorry. That's parent number 2. All right. Now I do the same thing that I did before. I'm just multiplying different letters together. I see a dominant gene and a dominant gene. I get big P, big P. That's a purple flower. I see a dominant gene and a recessive gene. Big P, little p. I see another purple flower, but it's heterozygous. Okay? I see this, little p, big P. I'm going to put the big one first, though. Okay? So I get big P, little p. It's heterozygous, but that's a purple flower. Uh-oh! Look at this. Little p, little p. I get 2 recessive alleles. That's going to be the white flower. So when Mendel crossed these purple kids together, right, in that second generation, what he got was he got essentially 75% of the offspring were purple. That encompasses this, this, and this. And then 25% were white. It's important to know the terms in order to set up the problems. Lots of students will tell me that they're okay running the Punnett square. It's the setup of the problem that confuses them. Remember this, in order to do the setup, you have to know these words, homozygous and heterozygous. These two parents up here are heterozygous. That means they have 2 different alleles. Homozygous, you could be homozygous dominant, and that would have 2 capital letters, or you could be homozygous recessive, okay? 2 little letters. In any genetics problem that you get, okay, the problem should tell you whether an individual is heterozygous, or homozygous dominant, or homozygous recessive, okay? That should be embedded in the problem, and you use those words to help you set up the Punnett square. And then once you set it up, running it, most students find that pretty easy. And then usually under the Punnett square, we write the phenotypes and the percentages for each one of the children. Okay, so I hope this makes sense to you. I hope that this goes from conceptual to practical in terms of setting up Punnett squares. Big things to know. Each of us has 2 genes for each one of our traits, but our sex cells, when they separate it out, we cut that in half, right? So that each one of them just has 1 gene for each trait. The Punnett square can show us how our sex cells can combine with someone else's sex cells, okay? And it can predict then what the offspring would look like. This happened in plants, but in class, we'll actually be able to look at some human traits and cross those up so you can see what your children might look like. Hope this made sense, and I'll see you on the other side. Thanks for listening.